bacteriophage, then finally to yaks or backs. Now let's first talk about the plasmid vectors. So this is the first one of our interest, plasmid vector. Now, plasmid vectors are very small size. The advantage of the plasmid vectors, we all know what plasmid is, so I'm not going to talk about the structure of plasmid in detail. So it must contain this origin of replication. So this is the origin of replication. First criteria fulfilled. Second thing is that it must have the selective marker. So it is ampicillin resistant marker is there. Third thing is the multiple cloning site or polylinker it is having here. So it is having these three important parts which are important for being a vector, and it is a pretty good vector. Why? Because it is having a high copy number, so it can be present inside the cell in higher amounts at a time. So when we clone our desired gene using plasmids, we can be ensured that the number of particular gene of our interest can be increased after few divisions inside the cell. Second thing is that it can have the visual marker for selecting the bacteria carrying the particular plasmid. So we can use this as a visual marker, because direct addition of this plasmids of this insert into this plasmids can give us any kind of biochemical expression or color change in the medium or something which can be visualized directly. We don't need to do any genetic analysis using that to confirm whether the clone is done or not. Okay, by looking at the colonies we can tell yes, the cloning is done or not. So it is very advantageous for that purpose. Time pretty less time consuming. Third thing is that also the promoters are present, and the promoters that are present in these plasmid vectors can also be introduced or utilized in vitro transcription studies. So we don't need to modify this for the in vitro transcription. We can use these plasmids, we can extract the plasmids out, and we can just do the in vitro transcription studies which will be required in the future uh, parts of the RDT processes. Okay, so here is the picture of how plasmid vectors are utilized for cloning. So this is our plasmid vector, this is our gene of interest, this red color thing. So what we'll do, we utilize the same restriction enzyme to cut those things out. EcoR1 are utilized here. EcoR1 is added, and it will cleave this segment of the plasmid vector, as well as it will cleave our desired gene. Then we add all these things together and we DNA ligase, which will fuse our products, which will fuse our vector as well as our insert, and vector insert are fused to produce a recombinant DNA molecule. Now we'll take this DNA molecule and insert into the bacterial host via the normal transformation process. So it's just the rising electroporation or any kind of calcium chloride mediated competence generation. It will easily enter into the bacterial cell, then it will start dividing and make a clone of our particular gene. Then we can extract it and do the further studies. Okay, so this is the mechanism of plasmid utilization. We have already discussed it, so don't need to talk about that. Now some new features about modern plasmids. Now in future case, what you've seen, it must have small size, origin of replication, must have multiple cloning site, and also selectable markers. We have seen all these things. But now what we can see, nowadays some important features, some new features must be there to recognize them as important vectors. The features are there. Some are expression vectors and have sequence that allow the RNA polymerase to transcribe genes. So sometimes they must act as both cloning vectors as well as expression vectors. What are expression vectors? We'll be seeing later. Now they can act as both cloning vector as well as expression vectors, and must have the sequence that allow the RNA polymerase to transcribe those genes and then can translate those genes into protein products. And also they must have the DNA sequencing primers in them. So in nowadays these two criteria are also being maintained to make a good vector. Now what are the major limitation of utilizing plasmids as a cloning vector? The upper limit of the clone is maximum 10 to 12 kb. So it's a very short segment of gene that can only be cloned utilizing this type of plasmids. Now it requires the preparation of competent host cell to transfer this particular plasmid inside the bacterial cell, right? So it is a pretty tedious job. Again, we need to treat the cell with calcium chloride solution and all these things. Set of protocols must be carried out before transferring our desired plasmid onto the bacterial cell. So it's a pretty hectic there. Third thing is that inefficient for the generation of the genomic libraries. Why? Because there are overlapping regions needed to place in proper sequence. So we must not use this kind of plasmids usually for making genomic libraries. And also for the genomic libraries, there are crude extracts of genes, so plasmids are not good choice for them. and obviously the preference are for only for smaller clones. We cannot utilize them for utilization of large clones. So this is again same type of thing in both the case. And it is an expression vector. There are often limitations regarding the eukaryotic protein expression. So most of the case we cannot utilize these plasmids for the eukaryotic protein expression, because most of the time the plasmids are acting as the cloning vector only, not as the expression vector. That's why nowadays plasmids are modified in such a way that they can act as both cloning as well as the expression vector, so that we can use them in the eukaryotic protein expression.